Together with first author Guru Shankar Chandramouli and other collaborators, Dr. Pomerantz's team started by investigating one very unusual polymerase called polymerase theta. Of the 14 DNA polymerases in mammalian cells, only 3 do the bulk of the work of duplicating the entire genome to prepare for cell division. The remaining 11 are mostly involved in detecting and making repairs when there's a break or error in the DNA strands. Polymerase theta repairs DNA, but is very error-prone. It makes many errors or mutations. The researchers therefore noticed that some of polymerase theta's bad qualities were ones it shared with another cellular machine, albeit one more common in viruses, the reverse transcriptase. Like Pol theta, HIV reverse transcriptase acts as a DNA polymerase, but can also bind RNA and write RNA back into a DNA strand. In a series of elegant experiments, the researchers tested polymerase theta against the reverse transcriptase from HIV, which is one of the best studied of its kind. They showed that polymerase theta was capable of converting RNA messages into DNA, which it did as well as HIV reverse transcriptase, and it actually did a better job than when duplicating DNA to DNA. Polymerase theta was more efficient and introduced fewer errors when using an RNA template to write DNA messages than when duplicating DNA into DNA, suggesting that this function could be its primary purpose in the cell. The group collaborated with Dr. Xiao Jing S. Chen's lab at USC and used X-ray crystallography to define the structure and found that this molecule was able to change shape in order to accommodate the more bulky RNA molecule, a feature unique among polymerases. Our research suggests that polymerase theta's main function is to act as a reverse transcriptase, says Dr. Pomerantz. In healthy cells, the purpose of this molecule may be toward RNA-mediated DNA repair. In unhealthy cells, such as cancer cells, polymerase theta is highly expressed and promotes cancer cell growth and drug resistance. It will be exciting to further understand how polymerase theta's activity on RNA contributes to DNA repair and cancer cell proliferation.